The first cell center. I've been collecting human samples on patients with pre-leukemia and acute myeloid leukemia since 1984. We now have over 60,000 samples, some of them serially collected on the same patients as the disease progressed from a pre-leukemic stage to a leukemic stage. This is a very precious tissue repository, and now the technology is available, so we would like to go ahead and start a serious interrogation of these samples. The idea is that we should now look at patients as they are progressing through their disease using all the technologies available to study DNA, RNA, protein markers, which would help us identify the earlier and earlier footprints of the disease. At the same time, we want to develop implantable devices and imaging devices. We want to develop vaccines to prevent cancer from expanding. So we have a 5-year plan during which we propose to identify first the biomarkers using the tissue repository. These biomarkers could be pieces of DNA, RNA, or protein, or any combination of these. And then in the later part of the 5-year plan, we start utilizing these biomarkers that have been developed for actually prospectively evaluating patients and then taking them back to a healthy population to see if we can identify through germline sequencing what individuals are at high risk of getting these diseases. And once we have those markers identified, if such markers exist, then how many of them are already showing early markers of disease, and how can we employ the latest evolving immune therapies and technologic advances to eliminate at the earliest inception any traces of cancer. So the whole idea is study the tissue repository, develop the imaging and implantable devices, use these two techniques to go back to healthy populations, start monitoring healthy individuals for the appearance of the first cell. This is what the whole concept of the first cell center is.